Anyway, so as Bob said, theme 3C is this trophic connection between coastal ocean and the kelp forest. I am going to be talking about the coastal ocean, or basically yeah, basically I would call my research sort of LTER adjacent. And it was, again, good to sort of see that, you know, basically most of the work is focused on the kelp forest. The kelp itself forest appears to be, from what I understand, smaller than one grid cell in my model, which is basically, this is a 1 kilometer model. And so anything that's less than the size of a kilometer or less isn't going to be resolved, which means basically all of the, you know, the very near shore dynamics. So what I'm looking at is basically the physical mechanisms that are driving nitrate transport and how that specifically controls phytoplankton bloom in the Santa Barbara Channel, but the whole basically channel wide. So I'm looking at dynamics on a mesoscale, which is basically greater than a kilometer. And I think that, you know, this sort of as looking at how, you know, the channel wide dynamics, offshore dynamics are defined, and then maybe, you know, then the next steps would be, this is sort of the first step if I were, you know, hopefully to continue then to look at how, you know, or what is, or if there is a connection between the offshore and the onshore. And this is sort of kind of trying to explain what the offshore is. Okay, let's see if it'll let me. Okay, here's the next one. Can I hide this? Okay, that's better. So the primary, so the basis of my results are all with, and probably a lot of you have seen this, but I'm going to go over it again, is a 3-dimensional ocean circulation model. So this is a regional ocean modeling system that was applied to the Southern California Bight. A lot of papers have been produced on this data set. And so basically it was created at UCLA in the Jim McWilliams group with Charles Dong taking the lead. And so we have here stored basically the results of the entire Southern California Bight, the whole domain. And it's stored in 6 hour average offline solutions. So it's a 1 kilometer horizontal grid and has 40 vertical levels. And the one thing to, you know, enforce that even though it has 40 vertical levels, this means though it does have realistic bathymetry. So that those vertical levels actually change. So they stretch to the size of the how deep it is at that point. So it is realistic in 3 dimensions as opposed to just 2. Okay, so this is the methods to try to understand, basically quantify nitrate transport and then see how it is related to chlorophyll, surface chlorophyll concentrations. So this study, first of all, because it the focus right now is seeing when phytoplankton blooms happen is the euphotic zone. So all of the results I'll be showing you is for basically the top 25 to 30 meters of the water column. So the ocean circulation model has the 2 major outputs that we're using are temperature, which it produces, and flow field. So with the temperature, I am doing 2 things. One is I am going to estimate nutrients using a temperature nitrate relationship. And then second, using actually those distributions or concentration of nitrate to identifying different upwelling mechanisms. The other thing is the flow field. So the flow fields are used in this case to also identify eddy dynamics for the recirculation that happens in the channel. That can give you presence, location, size, and lifetime, all the things you would want to know about the eddy. For the flow fields are also force a reverse particle tracking model. And this actually allows us to identify residence times throughout the channel, as well as where the water was originally sourced, like whether it was coming from the north or from the south. And then to identify regulators, these results are compared in a bunch of different ways to surface chlorophyll concentrations. And those come from satellite data that I got from Maddie, I think Kahu's group at UC San Diego. Okay, so first in estimating nutrient availability. So in this case, this is not a phytoplankton model. It is purely, I wanted to know what like the maximum available nitrate would be as it's upwelled, or, you know, as basically as it is transported into the euphotic zone. So mostly using the Plumes and Blooms program data and some Cal Coffee data, I was able to do a relationship for samples that were basically greater than 30 meters. So I wanted samples that might, you know, that where nitrate wouldn't have been taken up by, you know, phytoplankton or other things. So we get a pretty good fit between the nitrate, and actually these samples are actually nitrate plus nitrite, but I sort of lump them together to nitrate. And I have to thank both Jordan and I think Tom, who I saw that they had done a relationship, nice relationship in their paper about the kelp, and that also gave me the idea to do this with the temperature from the model. So it's a pretty good fit with temperature, nitrate, and then we can basically use the temperature from the model, which is in 3 dimensions, and translate into nitrate concentrations. So here's the first set of results, and these are just like a long-term average. So to give an overview, the left top is surface chlorophyll from the satellite data averaged over the 10 years of our study. I've removed the very nearshore values because I looked at actually multiple data sets, and there's a lot of discrepancies in the nearshore. And so basically I'm looking at the offshore, the deepest part of the channel. Below is the average flow fields from the model, which you can see that the west end of the channel is dominated by recirculation, or the Santa Barbara Channel eddy. And then to the right, top, we have the nitrate concentrations. And so this is going to look a little different than a lot of other papers where averages were looked at, but those are using sea surface temperature. And because this includes the whole, the euphotic zone all the way down to 30 meters, you can see that there's sort of 2 patterns, which is one is nitrate along the coast, and also nitrate in the vicinity of the recirculation. The last one is the residence times in days, also calculated using the reverse tracking. And this shows that there are, though I have this, the channel ended, but just in general, the whole channel, basically primarily because of the recirculation, shows elevated residence times, meaning, above about 7 days for primarily, and are particularly high in the central part. So in order to identify the upwelling mechanisms, there was I tried a lot of different things, trying to use the flow fields, but it's just hard because a lot of these things happen at the same time. I mean, you get upwelling and eddy circulation. And so what actually ended up being the sort of best way to see the differences was actually applying EOF to the horizontal nitrate concentration. So basically I took these depth averaged over the euphotic zone, these horizontal nitrate concentrations for the whole 10 years, ran EOF, and it gave me the different modes. And so the first mode turned out to be extremely dominant, basically contained 83.1% of the variability. And this I associated is most closely associated with wind-driven Ekman upwelling. And we can see that there are high concentrations. I mean, this is the it's not in concentrations because it's the EOF, the modes, but basically the positive values represent higher nitrate concentrations. And so we see them along the northern coast. The second mode, which was 5.8%, is basically advection into the channel from the west, coming from the west and advecting eastward along the southern California Santa Barbara Channel. So anyway, so that represents that. And then the smallest mode, or mode 3, which is 3%, really focuses on the pattern made by the eddy circulation. And this could be either the eddy trapping, upwelled water from that has come in, been upwelled another way, or a method called eddy pumping, which is where the eddy itself, winds up and basically uplifts with uplifted isopycnals, brings nutrient-rich water to the surface. So the mean annual cycle. So this is a comparison, and a lot of people also have, I'm sure, seen these kind of patterns. So the top one is the mean chlorophyll, and in this case, I've averaged horizontally over the channel. And there's a strong seasonal signal where you have high levels of chlorophyll in the spring. Basically, the chlorophyll is dominated by that. And the model also predicts that you would get mean nitrate elevated also at the same time. Again, remember nitrate being a function of temperature. We see that this is also similar to temperature, but it mimics that pretty well. So then the other ones are looking at the different principal components. So we have mode 1 principal component, which is wind-driven upwelling, and that is positive, which means when it's happening, pretty much simultaneously as the other spring peaks. So we see that and also, because this is more dominant, the values themselves are much larger than the other principal component. So we see basically starting in March and ending towards the end of June, we have wind-driven upwelling. Second is the principal component 2, which is advection into the South Channel. And this pattern is a little more irregular, but the highest values happen at the same time as wind-driven upwelling, which is not surprising because often you're going to get upwelling that happens not on the north coast, but is also happening, around Point Conception and upward that could be being advected in. And so that happens basically about the biggest values pretty much simultaneously. However, there are some other peaks and some things that happen where we see some, slightly higher, it's going to be higher concentration nitrate water coming in the winter. The last one is the eddy trapping pumping pattern. And this one is a little different. Again, we do see some overlap with the principal component 1 and 2, which again shows that all these things can be happening at the same time. And it's not surprising that a lot of the times when, I'll show a movie at the very end where you can sort of see the advection coming, the advected water of the wind-driven upwelling coming in, and then as it relaxes, the eddy kind of picks it up and sort of keeps it up. But what's interesting too is that there is some eddy trapping and pumping that happens basically during the summer and early fall. It's not nearly as strong as the other, as wind-driven and stuff, but there is that pattern showing up. Okay, so then the other factor that we looked at was the looking at where this water is coming from. And so basically by tracking, so we released, this was both with the residence times as well. Basically, I released particles on a 1 kilometer grid throughout the euphotic zone. So basically every 5 meters depth down to 30 meters, and track them backward in time for 60 days. And for residence time, actually, we basically just looked at how long that particle was in the euphotic zone before it it's actually basically from the time it came into the euphotic zone, and yeah, so but backward in time. And then this case, we actually looked at the maximum depth. So when we looked at reverse tracking, we wanted to see like, like where this particle upwelled from. So if it was like, 50 meters, 100 meters deep, that maximum depth is where the source was. And so here we have the annual cycle, and we have 4 different zones. We have the north, which is coming from the north or the California current. We have blue, which would be water coming from the south, which is Southern California countercurrent derived. Whether it came from the channel itself after 60 days was still there, which turned out to be not very many. And then if it upwelled in this sort of south of the channel, I'm calling other, because it's hard to determine exactly what the origin was in that case. So, but if you look at the pattern, you can see that the water coming from the north is dominant, is the largest, the same time as we're seeing the highest mean surface chlorophyll and mean nitrate, and is associated with wind-driven upwelling. And there's not much coming. There's this sort of other group is there, but otherwise it's dominantly. So this actually, this figure shows the fraction if I were to take the whole euphotic zone of the channel, this is basically the fraction of that water that's come from these different sources. And so it dominantly comes from the south, and some from this other category, but that really, the north, coming from the north of the California current peaks at the same time as the wind-driven upwelling, which is what you would expect. So next we looked at the eddy in terms of its dynamics. And this was a little bit of a surprise, and I had no idea what to expect because I don't think anybody's looked at this. But first, what I did is I calculated whether the eddy was present and then how big it was in terms of area. And I would say at the largest, at this sort of 400 or 500 kilometers, this is about the largest recirculation can get, because it basically fills the whole channel. So the eddy area was very variable, and the eddy was a bit larger in the summer and early fall, but the seasonal cycle definitely did not correlate with chlorophyll or any of the other, nitrate or any of the other parameters that we saw. And also these really large, so this is the average plus 1 standard deviation. And so there's actually a lot of variability. So even though this is an average value, it basically is showing that the eddy basically grows and gets larger and smaller, sort of just constantly on like I don't have the data shown here, but about every, I don't know, maybe about once a week, it basically just changes in size. So it's constantly changing, and also is present. I think I calculated that on average, 97% of the time you can detect some sort of eddy or recirculation. So the mean residence time in the Santa Barbara Channel, so that was calculated at 7.6 days, and I think that's what I have here for this dotted line. From what I've been reading, basically this residence time is important because if you have upwelling and the nutrients are basically transported offshore very quickly, then there isn't time for blooms to form. And so, or the chlorophyll concentrations to be elevated. And so what basically this eddy circulation is doing is it's causing this average residence time that is about definitely long enough for blooms when nutrients are available to form. And so, and that's pretty consistent. It's consistent basically there's no seasonal annual cycle. It's consistent through the whole year. And then on the bottom, I just wanted to show that then we calculated the mean residence time within the eddy. And that is actually even higher than the 7 days. So within the eddy itself, it averages 12.6 days, which is definitely high enough. And that dynamic is, contributing overall to this sort of higher residence time, sort of atypically high residence time, I think, for the Bight compared to other areas. So interannual variation. So this is in progress. I wanted to show just a few things. The first is time series of chlorophyll, surface chlorophyll average as well, which is, over our 10 years in particular, it's really varies a lot. There's much higher bloom some years, and a lot of them, the second one is the nitrate concentration calculated by the model. The third panel is the principal component for those specific years. And the bottom is the source zone. So this is, showing that the water from the N, which is the north, or California current water, is yellow, and from the Southern California, the countercurrent would be blue, and then the red, and the red is, again, this sort of unable to identify area. So at first looking at it, it's hard to sort of see a pattern, but there is a pretty, the other thing I wanted to mention too is that I was not able to correlate the strength of the upwelling using the principal components. And I think this is because although the temperature of the model, I did a lot of calibration where I compared, or validation. So I compared basically the model temperature to model time series, mostly from the LTER. And there was about a degree error on average. From 98 to about 2004, that error is pretty similar. But from 2005 to 2007, the model is much warmer than it should be. So it's predicting higher temperatures, which means that because the nitrate is higher with lower temperatures, it's going to underpredict the upwelling. So I wasn't able to so I ended up basically comparing each year individually as a pattern, as opposed to that relative strength. I mean, there's some extremes like in 99 that you can identify, but it's sort of hard to tell from year to year. However, one thing though that is clear is that on every year, even in 98, which is an El Nino year, that there is principal component is positive in the spring. It's positive in the spring, at least shows one peak every single year. And with that is correlated is water coming in from the north, or from the California current. So there's definitely this spring upwelling, no matter sort of what the year is, there is definitely a strong component of spring upwelling, and that correlates with high surface chlorophyll concentrations, and also with wind-driven upwelling. So again, I'm sort of working on how best to, sort of quantitatively like compare these years, and what else can we say besides the fact that there's a strong spring peak, and what could be driving the other things. But I wanted to just go into detail about one year, 2001. Now, this is what I would consider a year that looks most like an average year. So most like those annual cycles that we see. And I'm going to show a movie. So this is the nitrate concentrations over time. I'm going to start in the end of March and go through, I think, September. So what the first panel on the bottom is so first this is surface nitrate concentrations that are averaged vertically over the euphotic zone. The bottom is the middle panel is basically your principal component. So the red line, and I actually just cut it off because I wanted to be able to show some of these other things, but it does continue on to a peak. The red line shows wind-driven, the pattern that is most likely wind-driven upwelling. The green is advection into the southern channel from the west. And the blue is the eddy circulation. And in the bottom, we're also showing the source location, which I didn't write anywhere. But again, this is the source location. So this is the fraction of the channel that contains water that's either from the north, from the south, Southern California current, from within the channel, and again, the red from sort of unclear where it's from. So I'm going to start this movie. And you can see this is a typical upwelling event where we're seeing very high concentrations of nitrate that are flowing into the channel, initially along the northern coast. They're highest along the northern coast, but again, it fills the channel at some point. You can already see a little bit of some recirculation. So this would be basically the upwelling. And then at some point, the wind relaxation starts and you start to see the upwelling basically relax. And at this point, I'll stop it for a minute. What happens is that the higher nitrate water then basically gets retained in the channel by the eddy. You can see that it sort of moves around with the eddy. But again, sort of changes in size until we get to about here. And now the wind-driven upwelling is really dropped, and it's starting to be dominated by this pattern. I say advection, but basically the highest concentrations being in the southwest area of the channel. So the other thing, so this is what we're seeing during spring upwelling when there's a huge, high concentrations of nitrates that would flux into the channel, and particularly into the LTER and to the kelp beds over there. But as we go on, after that, even though it's not dominant enough to a lot of the times cause a phytoplankton bloom, you still see there's a lot of this sort of elevated nitrate, again, moving around. And the model is then identifying this as PC1, which is wind-driven upwelling. It's basically identified by high concentrations along the northern coast. But this is kind of I think that this might be a different mechanism, because there is not a lot of water at this point in the channel coming from the north. And you're seeing these high concentrations right along the coast. And so even though it might not be sort of, actually, again, this is sort of with me thinking from listening to all these, talks that have been given, is that this could be a sort of summertime maybe mechanism for nitrate concentration from the offshore to the kelp beds, which is that, and you kind of see as we go on, that there's these periodic, would be another one coming up. We still have the eddy containing water. But then here again, I'll stop it. We see this elevation in nitrate concentrations right along the coast. And again here, and I need to look closer at it, but I don't think that this is what we call typical wind-driven upwelling. So I think there's maybe some sort of, and also the system is stratified at this point too, pretty highly stratified, which is, again, usually doesn't happen when you have wind-driven upwelling. So you have these periods where, and I think that one of the mechanisms that in a paper that I know Melanie Fewing was involved in, where she hypothesized that there could be an interaction between the wind and the stratification that can cause this upwelling. So even though I just wanted to point out that these other, there's these dominant principal component. There's another one right there. There's these dominant, sort of standard, the principal component is predicting these wind-driven upwelling events, that there are these coastal, again, sort of upwelling events, which are, again, sort of atypical or not, doesn't fall into sort of our category of what we think of as in terms of mesoscale processes that are happening in the channel. However, these usually do not result in a phytoplankton bloom. So for what I'm doing, I am not, or elevated chlorophyll concentrations, I am not I haven't been focusing and studying those particular ones. And I think that's the end of the movie. Okay, yep, that's it. Oh, no, that was not. I want to go to the next slide. Okay, so in summary, the modeling basically supports a lot of the hypothesis that, but people have been able to observe, which is that there's an annual spring-driven upwelling event that basically causes the strongest chlorophyll, highest chlorophyll concentrations or phytoplankton blooms in the channel. And this happens every single year. So the other thing that was hypothesized is, I mentioned that the eddy itself is, since it's present a lot of the time, that it itself could be, upwelling water to stimulate blooms. And in general, when you have this, when I've read a lot about really, really large eddies in the ocean that are like, hundreds of kilometers, is that they really sort of retain, they have like a whole environment inside of them that they retain and then can transport, many, many kilometers, long ways across the ocean. And I don't think of the eddy in the channel really as that anymore. I think that, although there be some times when it can retain water that's been upwelled, I think that probably it's important that it does retain water. It doesn't really exactly trap it. It sort of, it basically keeps it contained, I think, within the channel to cause, to basically makes the nitrate accessible by retaining it. And that, so I see that sort of trapping, but I don't really see what's called eddy pumping, which is where there's nothing else happening and the eddy is just spinning up and causing a bloom itself. I don't think that the eddy is stable enough to really, for long periods of time, to do that, or maybe big enough too. But on the other hand, I think that the recirculation basically creates an environment where if you didn't have the channels there, everything would basically just be, advected offshore, and it would not be nearly as productive as it is. So I'm writing this up in a paper that focuses on the mechanisms that drive these elevated chlorophyll. And the biology, Dylan has generously helped me, agreed to help me, keep me honest and help me with the biology for that. Because I really want this to be a paper that is, read by biologists and physical oceanographers. And in terms of the LTER, I think there's, a lot to look at in terms of what could be coming from the offshore in terms of nitrate transport to the kelp forests, and possibly also, the fact that I didn't mention as much, but that there also is quite a big gradient, and it seems like there's a lot more nitrate coming on the west, in the Western channel as opposed to the east. I don't know if, different things are seen when in terms of observations. I'm hoping I have some smaller scale circulation models. They're not in the summer, unfortunately, that less than a kilometer, but I'm hoping to, with some new data that I have, maybe create some smaller scale models of the channel that will be able to, see a little bit more what's happening in that 1 kilometer near the shore. And this, of course, is not funded yet. So I am working on, thinking about how I can translate that into that. And I think that's it. Yeah, and thanks for listening, everybody. And if you have any questions, please let me know.
Thanks, Rachel. I have some questions, but when
Okay.
people raise your hand if you have questions, I'll start off with one. I mean, I'm wondering, I was wondering, like, do you have an idea of what the average time period of the kind of anomalous summertime upwelling events that you were seeing along the coast?
Not yet, no. I will, I mean, I really want to get a paper out. So I'm writing up what I have right now. And then I did do some, like I tried to do some focusing on those, you know, those summertime anomalies. And it was not clear to me what was going on. So I would probably need to really focus on that. I mean, I remember seeing it and thinking like, I should let Bob know about this. But I haven't really spent a lot of time. My hope is that, well, I'm hoping to get this paper submitted by June. And then after that, you know, again, I need to write a proposal because I really want to spend more time on it. So, but that would be one of the things that could be, you know, of interest. And also if I could, you know, it would be nice to be able to do it and look at it at a smaller scale as well. So that's as far as I've got. The movie is about as far as I've got.
Okay. Jen and Allison have questions. You want to start, Jen?
Hi, Jen.
Hi, Rachel. That was awesome.
Oh, thanks.
Nice to see you again. It's been a while.
Yeah.
I have sort of a quick question, which is that I noticed that you have those offline solutions from McWilliams through 07. Is there any more recent movement on that?
Yeah. So I also have another, which I actually used for some modeling in a paper that Dylan wrote and did. So I have now another data set. It overlaps a bit, but it runs from 2004 to 2013. So it's still not, you know, super, you know, it's not super, you know, recent, but it does extend the time. But what's nice about that data set though, is that, so this one, I'm missing some data from it, meaning that there is they didn't give, when Satoshi did this, you know, now seems like very, very long time ago. They didn't get any of the wind data that drove the model or any of the surface boundary conditions. And so the nice thing about this other data set is that it'll extend the years, but it also has surface. So I can do things like look a little more details, either do some, you know, nesting and creating smaller sub-scale models, or also possibly looking at, you know, again, one of the things that is important about these summertime ones is that what kind of wind is driving it? Like, you know, whether this is sort of like your standard, you know, like I do have a little bit of wind data from this time from another model that I've been looking at, and it's kind of erratic, but it does show that, you know, the wind stress goes way up when you get the sort of large spring, you know, blooms and spring upwelling. And that doesn't happen in the summer. The wind stress is really erratic. But like one of the things as I was doing this, I was thinking like, I could at least dig through that data and see if there's any connection. I mean, there has to be some connection with the wind, but like whether I can see that, you know, change happening. And so I would love to have more data. It just, for years I've been trying to, you know, get funding to get somebody else to do it for me. I think I don't want to promise anything, but I feel like I need to finally do this just myself and figure out how to create more years. But I do have up to 2013, and it actually is a much bigger area. It covers not just the Southern California bight, but it covers all the way up to Morro Bay. up the coast and around Point Conception and also offshore pretty far. So
Cool, thanks.
that's available. Let's see, I can't see, is there any?
Allison has a
Oh,
question.
Yeah.
Hi, Allison.
Thanks for your talk, Rachel. I have
Oop,
two.
that was not the, why did that happen? Okay.
I have two questions. One is
Okay.
a dumb question about EOFs. So you said that the EOF3, the Eddy mode, accounted
Right.
that 3%. The way to think about the 3% is it accounts for 3% of the variability.
Yes.
That's not the same as saying that it is 3% of the time. So
Right.
So if you had to characterize, sorry if you presented this number, like what percent of the time is the
Well, I'm trying to think, do I have this here? I mean, if you look at, again, this is the annual cycle. I mean, even though it's smaller, the reason why it could be relevant is that, you know, the principal component one, although it is, you know, again, it's much stronger and has more of the variability, it only happens sometime, you know, during the spring, really. You don't see that as much. And so during these times when the principal component is negative, the principal component one, then these other ones, if you're going to see transport, are going to be what's driving it, basically. So, you know, because the Eddy is, you know, I mean, I would say, you know, the Eddy could be important for 6 months even, you know. I don't know exactly, I'd have to go back and look and calculate it, but it's not just also time, it's also strength. I mean, sometimes it's higher than others. And, you know, I mean, that's sort of why for this figure, I was trying to show that, you know, if you were to plot it with the full principal component, actually, you could also see it here, but if you were to just, if I were to put the full strength of principal component one on here, you couldn't even see the other ones, right? And so, but when this is gone, you know, these other things are, even though they might not be creating, you know, these enough to like, you know, when you get doing long time average, where you're looking over like that annual cycle, the average, they sort of disappear. But if you're actually looking sort of in real time, they might be small, but they could be important, especially if like at a place like the LTR, I mean, that's actually percentage wise also a very small percentage of the actual area, right, in the channel. So, yeah, so I mean, again, I don't know percentage, like what, you know, what that would be, but it, you know, I don't think it's insignificant. I mean, I think it's, you know, more, I mean, it's more than just 3% of the time, is what I should say.
I have a second question, but I want to give someone else a chance to ask a question too.
Do I see any other?
I have more questions, but I don't think anybody else raised their hand. So go for it, Allison.
Okay.
I missed what you said the vertical resolution of the model is, and if it's too coarse to be able to get stratification. Like, can you?
Yeah, so I didn't, that started again. Okay. Yeah, so I actually did calculate stratification. It is not too coarse for that. I'm not showing it here because I just, yeah, that is a variable that I have calculated. So I have, yeah, see if I want to try I'm trying to see if I could try to pull it up, but I think I don't know if I'm techy enough to switch to show it. But anyway, but yeah, so we do a stratification. The stratification, it actually, you know, it follows the same cycle in that, I mean, inversely. So like what you see is the stratification is very low, you know, January through, and then towards the end of the upwelling season, it starts to grow and then becomes strong. So that's what was surprising to me, is that a lot of those, you know, summer kind of whatever we want to call them, the summer upwelling events happened when the system was stratified. And also, so basically the way that the model works is that every single point has 40 levels. So whether it's 10 meters deep or 20 meters deep or 1,000 meters deep. It's always 40 levels. But a lot of the in order to get like boundary layers and stuff like that. So what happens is that the layers stretch out, they're flexible. And so you have more at the top and the bottom, and then less in between. So it basically, it's like it's like a sort of, so the layers are not static, meaning like they're not the same, they're not like 10 meters deep through the whole channel. They change that way. So you actually get more resolution in the areas that are shallower, because there's going to be 40 levels as opposed to when the channel gets deeper. But the idea is that you have less, there's less changes happening, basically fluid, in terms of the circulation.
Thanks.
No problem.
Oh, Nick has a question. Go ahead, Nick.
That was great, Rachel. Thanks. This creates lots of interesting ideas. Do you get the same, have you looked at, do you get the same modes if you do the EOF analysis with things like, say, surface currents? My interpretation was that this was EOF based solely on nitrate patterns.
Yes,
right?
So I tried that and it was not clear. I mean, it was really, I tried doing EOF on the surface currents, or the depth average currents, and there just wasn't a strong, there was just so much variability. It was hard to see a pattern, basically. And I also tried other things, I tried to look at, I was looking at because isopycnals are important. So I was looking at changes in isopycnal depth and all that, and isotherms, and it just was, this was the nitrate EOF was the most consistent in terms of, being able to see patterns that are happening at the same time, like processes that are happening at the same time. It's really hard because I don't, when you have, advection coming in and you have circulation, you have eddy circulation and you have upwelling, I mean, it just gets really messy. And so it's hard. I mean, you can't, in some ways you probably can't exactly separate them, but that using the nitrate just ended up being the easiest way to kind of tell a story, basically.
Cool, thanks.
I was just wondering about the map that you showed of the residence time. It showed the residence time close to the coast where the kelp forest is.
Yeah, I was thinking about that.
Like over 12 days. And I'm wondering, is that so? And
yeah, I would say that's a little.
Is that the horizontal residence time as well as the residence time within the photic zone? Is it within the 1 kilometer grid as well?
Yeah, so it's within the 1 kilometer grid. There is a little bit. So I was thinking I don't, there are some issues with boundaries there, which means that sometimes things can, particles get stuck on the boundary, not unrealistically stuck on the boundary. So I'm not totally sure how, I mean, I definitely think that there are probably elevated residence times, sort of like the way we've seen, with some of the water cypera stuff, where we see that the coast, things are moving slower along the coast than they are farther out. So I think there's some of that. But I would say in that one grid cell right next to the shore, and I don't think, I wouldn't necessarily put a lot of, those, I mean, there's definitely some retention and stuff going on there, but that you'd need to look at a smaller scale model, basically, to really, tell more about it. I mean, I think there's something, I think it's something there. I think it is important, but this is not, the resolution is just too big to really tell you any sort of besides sort of qualitatively, to give you any, kind of numbers. I wouldn't trust any of those that are right there.
Okay.
Because of the inaccuracies in the model. Yeah.
Another quick question. We have, as you, I think we talked about this before, and I can't remember. Maybe you tried, but we have all this data on bottom temperature from all of our sites. That's high frequency. Would you expect that record to match up well with the modeled estimate?
Yeah, I mean, it's relative. I think that, so I did, actually, now that I think about it, I don't remember. So going back actually to this figure, the white dots are the LTER sampling sites, I think. And so I have a whole section in the paper that I've written where I do compare the temperature of the model to the temperature that's collected in the field. And I remember you giving me that bottom data. I don't think I used that. I think I just used the standard, LTER, the bottom temperature. The bottom temperature is more likely to be accurate because we don't have waves or anything. I mean, the surface is definitely a little iffy, like right at the surface, accuracy wise. So, I mean, basically you get about, like, on average, like I think I calculated root mean square error is about a degree, but it's the same for most of, so basically what you can get out of this more than actually like pointing to a specific temperature is the patterns. So what we want to know is whether the model is recreating the right patterns in terms of when the temperature goes up and when it goes down. And so the errors, what you hope for at the most is that the error is going to be the same for each year. So it's about the same, which is true until, again, you reach about 2005, and then it kind of goes off a little bit off. And so in terms of, like, it depends on what you're looking at. Like if you have a specific event, that you want to look at that's happening, you would need to compare the model to that time series. And you can see, not only just look at the difference, like, but also to see, like, is the pattern that you want to, that you're seeing with the model, is it happening with you're seeing in the field data, is it happening in the model? And if it's happening in the model, then, what are the patterns driving the three-dimensional temperature? So it's sort of used that way. I mean, there are times when, like, for example, the example I showed you in 2001, the movie I showed you, and that was, that one was like, it was very, very close, meaning the model really, like, hit the temperature right on the head for that, upwelling event. Other times of the year, model may be off for a few weeks. It might, it just kind of varies. And so you need to, I mean, I know Leonel Romero, who I've talked to a lot about this, is like, what you hope at most with the model is that statistically it's going to get it right. Mm-hmm. But it's not like, if you were to look at like one specific point in time, whether that is going to be reproduced, the odds are no. It won't be exactly right, but it should show you, like, what are the dominant mesoscale, patterns happening, both in terms of the temperature and the circulation, which is connected. So.
Great. Okay. Thanks, Rachel.
Good. So I think that's it.
We're pretty much out of time here.
Oh, we are. Okay. I didn't realize
A
that.
minute if anybody has another question, but otherwise, thanks everybody for coming. I see some yellow clapping hands.
Oh, thank you.
Great. Okay. Thanks.
Good.
Rachel.
Yeah, and if anybody has any questions, feel free to contact me. I'm trying to unshare. Now, how do I do this?
I definitely will. Thanks.
Okay.
So Rachel, I have a question for you.
Dan has.
Oh, wait.
Okay.
Stop share. Okay. Oops.
In your annual cycle that you showed,
yeah.
And I think you were kind of answering my question with Bob, but in your annual cycle, is that depth averaged over the photic zone for the whole channel? Is that
Yes.
what it is?
Yep.
And the seasonal cycle, I think the model gets it right, but you know,
Yeah.
the absolute values are actually quite higher, right? I mean, from it looked
For
like
which-
from your model, it looked like from summer through fall, you're averaging 3 to 4 micromolar nitrate. Whereas, you know, based on our bottom temperature measurements or bottle data in the kelp forest, it's less than 1.
Oh,
And the
interesting.
magnitude is really important because when it gets that low, it just doesn't do much for supporting
Yeah.
kelp, which is why we've started looking at other forms of nitrogen to support the kelp during the summer, because there's just not nitrate, you know, in the summer.
It's
Now
interesting.
there's some
So-
infusions of nitrate that come in, and maybe it's through some of these mechanisms that Melanie talked about, Melanie and Live
Right.
talked about. It
Right,
could be
right.
there's some short pulses of internal waves, or maybe some
Right. Now
forward-
this model cannot do internal waves. So this is not what I'm showing you is something else. So you're right. I've read those papers too with the internal waves or probably coastally trop waves or, you know, some sort of, you know, either internal waves forced by tide or forced by wind forcing. And that is, yeah, those are just not, they're too small, basically. And they're right near the coast. So basically, if the model is over predicting the nitrogen, the nitrate, then that would mean that it's also saying that it's colder, probably, I guess.
Yeah,
that
I
it
mean,
should be.
if you're generating nitrate from temperature, then I'm
Right,
wondering if that's
right,
because you're
right, right.
averaging over such a deep water column.
Right,
most of
right.
the
Now,
channel.
right. If I, like, for example, if, you know, I think a lot of the LTR sites are much shallower, is what I understand.
Oh yeah, seven.
Right. So if I were to then just average, I mean, I can average it over whatever. The reason why I have it averaged over the 30 meters is because because this paper in particular is looking at mechanisms for channel-wide blooms. That's what I want to do. But you could easily look at, you know, the top 10 meters and see if it does a better job.
Mhm.
Yeah. So,
But it may be what's deeper than that is what's driving, you know, the upwelled water is obviously deeper than 10 meters. So,
right.
Right. But-
Yeah.
but for inshore, you know, it's it is only 10 meters deep where the kelp, most of the kelp is. So,
Right. And so
um,
you'd have to look at, like, you know, like I would look at, like if I were interested in that, I would start looking at cross sections, like, you know, along the coast and sort of see like what's happening in terms of, like, if this is happening when the system is stratified, you know, you look at, like, you know, some field data of like what the stratification looks at and how accurate the model is that way. And, you know, how that could affect transport, because it could be that the model may be getting some of it right, but not all of it right, you know?
and so what is the temperature data that goes into the model?
So the temperature, just like the currents, are produced by the boundary condition. So it's basically a scalar, it's a temperature transport model. So you have the model running that is that creates the circulation. And then you have a scalar transport, which is temperature, salinity are all done this way, which is basically coupled with the circulation. And so they're run together, because also you can get baroclinic effects where you have the temperature and the, you know, the temperature can, I mean, I don't know if this ever happens in the channel, but, you know, if there's enough of a temperature gradient, you can actually get currents, you know, created by the temperature gradients.
And is
So-
there much confidence in the model? And I know I should know this because we've used these data before for our kelp dispersion
Right.
stuff, but much confidence in the model as you get close to shore.
Less confidence.
Uh-huh.
Yeah. I mean, I would say, although, you know, I mean, all of the data that I compared the temperature to in the model was from the LTR sites. I mean, it was the bottom temperature I was using, you know, but I think I was using the bottom temperature. And I mean, it did, it did a pretty good job of modeling that.
Okay.
So I would say temperature is probably more likely to be accurate as opposed to circulation.
Oh, that's
In that,
interesting, because
you
our
know,
bottom
the-
temperatures are typically for the most part 15 degrees or higher
right,
during that period. And at 15 degrees, there's just not a lot of nitrate in the water.
right.
Certainly
right,
not
right.
3 or 4 micromolar.
Right. And it could be that these, these little upwelling events or whatever they are that happen in the summer, I mean, they may, even though it looks like they're near shore in my model, they may not be close enough. You know what I mean? I mean, they may not, it's unclear, like what happens between, that 1 or 2 kilometers that the model is basically missing. I mean, I would just assume that that's missing and that, you would need to look at, like what I would do, for example, if I was really wanted to, dive deeper into this, would be to create a smaller scale model, like, and like a sort of like basically embedded in what the model I have now and make a smaller scale model.
Yeah, and there are